Kate Volk, master's student in Jill Hamilton's lab, and she's going to talk to us about her thesis research. So welcome, and I want to let everybody know that we are recording. So go ahead, Kate, when you're ready.
All right, so thanks everybody for joining me. So my title has changed. We were talking about that earlier. So I'm going to be talking about the evolution of trait differences across a species range and the implications of that for grassland restoration. Whoop. All right, so I'm going to go over just a little bit about what grasslands are and why they're important, although from Jess's talk last week, we should all be in love with them by now. So just briefly, I'll mention that grasslands are found in temperate regions across the world, but in North America, we see them across the Midwest and into Canada. And so like the name suggests, these ecosystems are dominated by mostly grass species, but they also contain a beautiful diversity of flowering forbs. And you can kind of see that in this photo. They're scattered around in between some of those grasses. And so they have numerous ecosystem services, including being a pollinator food source, providing habitat to common species like the white-tailed deer, and they can also house rare and endangered species like the western prairie fringed orchid. And so besides offering a food source and habitat, a diverse grassland can also filter harmful agricultural byproducts from water. And then the expansive root system below ground can sequester carbon from the atmosphere. And so the benefits of a healthy grassland system are not short by any means, and I only talked about a few. However, despite their importance, grasslands are considered an endangered ecosystem. And this is due to things like urban expansion, invasive species, so introduced plants that aren't native, that entered grassland systems and took over both because they were planted and then sometimes accidentally introduced. And then because prairies have some of the greatest soils in the world, the largest cause for their decline is, of course, conversion to row crop agriculture. But because of their many benefits, we need to conserve grasslands, and we can conserve grasslands through management practices like prescribed burning, as well as grazing. However, we can also conserve grasslands by creating new ones through the process of restoration. And so restoration aims to bring back, sorry. So restoration aims to bring back species diversity, to recreate the systems that offer those valuable services we just chatted about. However, there are some challenges when it comes to sourcing seed for grassland restoration. And a big one we need to mention is the sheer demand for good seed. Restoring land is really expensive in regards to both money and time. And so ensuring the seed that you have will be successful at the restoration is a huge priority. But without consideration of where that seed is coming from, you might see different sources of seed performing differently on the landscape. And so looking at this graphic representation, you can see that seed source 1 is a good match for the restoration site. Seed source 2 could be doing better. And we're just saying this is dead. Seed source 3 hasn't survived at all. And so a question that is frequently asked is, how do you know which seed source is best? And a lot of information says that local is best, and that's because of this idea of local advantage. And local advantage is where seed that's grown and has been growing in a location for many generations is optimized to the abiotic and biotic conditions of that location. So if you look to the figure, on the x-axis, you have two different environments, and the y-axis is the fitness response. And we can see that seed source from location 1 does better in location 1, and seed source 2 does better in location 2 when grown, does better in location 2. And so if you move that seed, you can see that there's a reduction in fitness. And so this idea is really well researched, and intrinsically it makes a lot of sense because plants can't move in the typical way an animal can. And so over time, they need to evolve to suit the conditions that they're being subjected to. However, there are some issues with defining locality, and one of them is simply that there isn't consistency of what local is across agencies that are implementing restoration. And so to some, local could mean just 1 mile, but to others, it could be an entire state. And so one way to define local is to create seed transfer zones. And so seed transfer zones are widely used in the forestry industry, but their practicality is spreading to grassland systems. However, creating these transfer zones is a really time-consuming process, and it requires years of research. And so some research has suggested that ecoregions, as shown in this photo, can be used as a proxy for seed transfer zones. And so the idea here is that seed found growing in a defined ecoregion should only be moved within that defined region. And if seed is moved beyond that defined boundary, you can risk that that reduced fitness response. But however, maps like this are created with only a handful of species, and grasslands, like we saw, contain a bunch of species, and they all function differently. And so they have differences in their adaptability to conditions. Additionally, these ecoregions are created with the assumption that the climate is stable. And of course, we know that's not the case. So specifically in the Northern Great Plains region, it's predicted to have warmer temperatures, higher evaporation demand, and alongside those, increases in extremes of water availability, from excessive flooding, like you can see on the left of the screen, to prolonged periods of drought as depicted on the right. And so finding seed that can tolerate these drastic changes in water availability is increasingly important for restoration purposes, to ensure persistence on the landscape. And so balancing local adaptation while maintaining their adaptive potential to respond is becoming a requirement for long-term success and resiliency in our restored populations. And so with all that in mind, we arrive at a broad goal of this research, which is to evaluate how different mechanisms of selection can inform seed sourcing guidelines. And so a species can be distributed across a really large range, and we know that selection can occur across broad scales, such as climate. So if you look at the figure, you can see populations of some kind of echinacea species growing under different climates. So if you imagine from the left of the screen, it's California, and then to the right side of the screen, it's New York. And so across that range, populations on the left side might have adapted to drier conditions, and on the right, wetter conditions. And then in the middle, let's say there's an average amount of precipitation. So we can zoom into a smaller section of land where the climate is the same. We can find populations of echinacea growing across differences in the soil environment. And then specifically shown here is differences in the soil texture these plants are growing on. And so part of my research aims to understand how selection associated with climate can influence trait variation, but also how selection associated with those fine scale differences in the soil environment may impact these populations. And understand how populations are different, we use a trait-based approach, and we look at physiological traits that are involved in the balance of carbon uptake to water loss. And we do that because this balance is really crucial for plants to maximize their growth while minimizing mortality in any environment. And so we're going to cover both chapters of my thesis research and investigate both deal with investigating the influence of changing water availability on trait variation. And so the first chapter, we're going to test for trait differences across ecoregions while they're grown in a common garden. And then the second chapter, we're going to evaluate the role soil texture origin has on trait variation when we're manipulating the amount of water. All right, and so for both chapters, we are going to be looking at the perennial forb, GM triflorum, which is commonly known as prairie smoke. And so while prairie smoke isn't listed as a species of concern, it has a really important role on the landscape. And that's because prairie smoke is one of the first flowers that pops up on the prairie, and that makes it a really important pollinator food source when not a lot of other plants are blooming at the time. And additionally, prairie smoke has been shown documented across a broad range. making it a good model organism for my research questions. A little laggy. All right, so to start us off, we're going to investigate trait differentiation in prairie smoke from across different ecoregions while they're growing in a common garden. So from across much of the GM triiform's range, we harvested 19 populations. And so they're indicated with these symbols across the map. And so populations were harvested from 2 very different habitats and across 3 unique regions. So the first one, we of course see prairie smoke thriving in the prairie. And the prairie is characterized by deep, rich soils, and they receive moderate rainfall year round. But we also see prairie smoke growing in Manitoba alvars, as well as Great Lake alvars. And so alvar soils are a stark comparison to the deep, rich soils in the prairie because they have a very thin layer of soil over limestone. And so due to the lack of soil in these regions, alvars are subjected to shifting extremes in water availability, where every single spring the landscape is flooded, and every single summer the landscape is completely desiccated. And so seeds from across these 3 regions were planted into a common garden, which is located at the Regional Science Center in it's more like Linden, so it's not too far from here, and that's within the prairie environment. And so we decided to use a common garden because if seeds sourced from across different environments are planted all in one environment at the same time, when we're looking for differences, and if we find some, the differences that we see should largely be due to genetic differences because that environment is the same. And so to understand how these populations sourced from different regions are different, I measured both the carbon isotope composition and a couple stomatal traits. And so for the people in the audience that aren't plant people, stomata are tiny pores on the surfaces of leaves, and they're surrounded by special cells called guard cells. And so stomata act to regulate the gas exchanges between the plant and the atmosphere. And some of that exchange includes how much carbon is taken into the plant and how much water is lost. And so by definition, the ratio or the amount of water lost to carbon gained is called a plant's intrinsic water use efficiency. And the carbon isotope composition is the ratio of the C13 isotope taken into the plant to the C12 isotope taken into the plant. And so that ratio can be used as a proxy for the plant's water use efficiency. And so to measure the carbon isotope composition, this one was easy. We just sent ground leaf tissue to UC Davis and they could determine that ratio for us with their mass spectrometer. To measure stomatal traits, we could do that a little bit more locally, but we first had to create molds of our leaves using liquid bandage. And then once that mold was created, we photographed it for analysis, and the resulting photographs kind of look like the background of this slide, where you have a bunch of stomata sporadically throughout the image. And so on these images, we measured the guard cell length, and we also counted the number of stomata in the area of view and divided it by the area of view to determine the stomatal density. And so what do we expect to see? So due to the differences between the alvar and the prairie region, we made some predictions about how these traits might look. So while the prairie habitats do experience drought and flooding, it's fairly unpredictable, and the soils can act as a moisture buffer in some cases compared to that consistent, predictable fluctuation of water experienced on the alvars, where every single spring they're flooded, and then by summer they're experiencing drought. And so due to the differences in water availability between our regions, we expect to see strong differences in traits linked to water use. And so we predict that these differences will show alvar populations exhibiting a greater control over their water use compared to those prairie populations. And so in the Common Garden, we're specifically asking if those traits are differentiated by region. And then we also want to know if the amount of genetic variation is different for traits across regions. And then lastly, we want to know if there's a relationship between trait variation and then the climate of origin. And so for our first question, are these traits differentiated by region? We have some predictions. Again, because alvar habitats are experiencing those predictable extremes in water, from flooding in the spring to complete desiccation by summer, we expect they're going to have a greater control over their water use. And so specifically for stomatal traits and water use efficiency, we predict that alvars are going to have a smaller stomatal size, but a greater stomatal density relative to the prairie. And we think alvars will exhibit those characteristics because many but small stomata have been shown to respond faster to fluctuations in the environment, and they're definitely coming from a fluctuating environment. And then in addition to that, we expect alvars are going to have a greater water use efficiency relative to the prairie. All right, so looking at this graph, I'll just tell you now, it is water use efficiency is regionally differentiated. So on our x-axis, we have our 3 regions. So we have Great Lake Alvar, Manitoba Alvar, and then the prairie. And then on the y, we have our carbon isotope composition. And so less negative values of our carbon isotope composition correlate with a higher water use efficiency. And then a more negative value is going to indicate a lower water use efficiency. And so in the common garden, we can see that the Great Lake Alvars are exhibiting the greatest water use efficiency and the prairie have the lowest. And then the Manitoba is just kind of intermediate in that result. And then, so we can also look at our stomatal traits and we kind of find the same thing, that stomatal traits are regionally differentiated. So on the right side, we have our Gardasil length, and we see that both alvar regions this time had the smallest stomata relative to the prairie. And then on the left, we're looking at our stomatal density, and we can see that those alvar populations exhibited a greater stomatal density relative to the prairie. So what does it mean? It means that when alvars were moved into the prairie environment, they exhibited greater control over their water use by having smaller stomata and more numerous stomata relative to the prairie populations. But a reminder that these traits were collected in the common garden. So why are alvars exhibiting the greatest control over their water use in that prairie environment? And so to answer why alvars are retaining greater control over their water use, we need to investigate the amount of genetic variation in our traits. And to investigate the amount of genetic variation in our traits, we looked at narrow sense heritability and evolvability. And so narrow sense heritability is the amount of additive genetic variance in our traits divided by the total phenotypic variance, and it can range from 0 to 1. And so higher values of heritability indicate the trait exhibits strong genetic control, and lower values indicate that that trait is highly environmentally controlled. And then if we look at evolvability, that's where we're taking that additive genetic variance and we're standardizing it by our traits. And so evolvability is then the expected change in our traits per generation. And that can tell us how much variation present in a trait is available for selection to act on. And so when we're making predictions about how much genetic variation is in our traits and if it's different across regions, we can make some predictions again. And so as a reminder, and you're going to love these photos by the end of it because I'm going to show them a few more times, alvars have extremely predictable cycles of water stress, from flooding in the spring, and if I was in person, I'd maybe have you say it with me, and then by the summer, they're experiencing drought. And again, while prairies do experience these extremes in water availability, they are more stochastic over time. And so why is predictability important for the amount of genetic variation? So in this case, predictability in water stress can optimize traits for those conditions. And so with that optimization, it can increase the chance that trait is going to be passed to the next generation. And so it's going to increase our trait heritability. But however, because of this optimization, total trait variation can be reduced. And so we expect that because of that predictability and stress, we think that alvars are going to have an increased trait heritability, but a decreased evolvability relative to those prairie populations. And so did we find differences in heritability? Just looking at this graph, we can see that heritability is similar, even if you're being tricked. That variance is saying there's no difference between heritabilities for guard cell length. So the amount of genetic variation was not different across regions. However, you can see that these values are pretty low to intermediate, right? Because heritability can range from 0 to 1. And so this is important because the amount of genetic variation tells us why alvars are retaining greater control over their water use when moved to the prairie, and the amount of environmental control indicates that traits can also be determined by the environment that they're being moved to. And so this plasticity may be extremely important because it indicates some potential to withstand future changes, such as climate change. If we look at the evolvabilities of guard cell length, we again find that the values are similar across the region. But what really is important here is that the values are extremely low. So they range from 0 to 0.1. And so if we think again about what evolvability is, or the amount of variation present in a trait that can respond to selection, our values indicate that these traits don't have a large capacity to respond to selection. So what does that mean? Well, it means populations could experience an adaptation lag under changes in climate. However, if the plasticity that we see in our estimate of heritability, if that plasticity is not costly to the plants, plants may not need to adapt, but rather just use their plasticity to respond to the changes they're undergoing. And so this last question, is there a relationship between trait variation and climate of origin, is really to understand why we are seeing trait differences and where some of the genetic variation we saw in our traits is coming from. And again, we believe the selection associated with differences in water availability between our regions is responsible for trait differences. And so to answer this question, we downloaded 30-year climate data. So it was average 30-year climate data, and we conducted a PCA. And so what we found was PC1 explained roughly 47%, and it was primarily loaded by mean annual temperature and degree days below 18. And then PC2 explained roughly 30%, and its highest loadings were the climate moisture deficit and the annual heat moisture index. So PC1 is primarily temperature based, and PC2 is primarily the precipitation based, sorry. And so with this data, we ran linear regressions with combinations of PC1 and PC2 as predictor variables. And across all traits, we found that PC2 had the greatest model fits. And so the subsequent results are going to show only the relationship of PC2 and our traits. So up first is water use efficiency. So on the x-axis, we have our principal component predicting our trait response. And along PC2, we see that larger values represent a greater climate moisture deficit and annual heat moisture index. And so large values are showing that there's less water available. And so, and then over here, we have our shapes representing our populations, and their symbols represent the region that they're coming from. And so looking at the graph, we find that as conditions become drier, water use efficiency is lower. So we found that there was a relationship between climate and water use efficiency. And we look again at our stomatal traits on the right, we can find that under drier conditions, our size of our stomata is increasing, and our brain fart, sorry. And there's going to be less stomata on the leaves under those drier conditions. So again, we see that there is a relationship between our traits and the climate of origin. And so circling back to why we asked this question, we can see that the genetic variation we found in our traits can partly be explained because of the source environment of our populations, and it's specifically related to that water availability. All right, so some conclusions for that chapter. Understanding how the source of our seed influences trait response when it's transferred is really important consideration for grassland restoration. And we found that region of origin contributes to genetic differences in traits important for the balance of carbon uptake and water loss. However, when we compared the amount of genetic variation in traits across regions, we did not find differences in their heritabilities. Rather, we observed low to intermediate values of narrow sense heritability. So lower values are indicating that traits are more likely to be shaped by the environment they are in, suggesting that these traits are to some degree plastic. And that's an important consideration for restoration, as it can tell us how risky our seed transfer is going to be. And so under climate change, that plasticity may be an important way plants can mitigate shifts in their environment. However, if the cost of such plasticity is too high, these populations may experience an adaptation lag because the expected change in those traits per generation or their evolvability was really low. And then lastly, we determined that climate of origin, specifically variables related to water availability on the landscape, contributes to the genetic differences we did find. And so this tells us that selection associated with climate across our ecoregions contributed to the evolution of the differences we saw. And this is an important understanding for the potential movement across those regions, like we talked about in the beginning. I'm gonna just take a little. All right, so we just looked at how selection associated with climate can impact trait differences. And so now we're gonna look at how selection associated with fine-scale differences in the soil environment can impact traits. And we're still interested in the influence of changing water availability on trait variation, but this time we're manipulating it. So for this experiment, we chose just 6 populations, and they're all shown on this map. The 6 populations we sampled was in 2019, and it was across just a small range in the tri-state region. And so across that small range, there's really few differences in climate. And while climate was highly similar across the sampling range, the soil and environment prairie smoke populations were growing on was different. And so using population coordinates, we extracted properties of that soil environment from the NRCS Web Soil Survey and chose the 6 populations based on soil texture. And so as you can see by the textural triangle, 3 populations shown in the red gradient are sourced from distinct sand-dominated sites, and then the blue gradient populations were sourced from loam soils. And so we chose sand and loam as our soil textures of interest due to their contrasting water holding capacities. And so that's saying like sand particles do not store water. And therefore populations growing on sand soil textures do not receive as much water. And that's a comparison to loam particles, which allow both the storage and release of water for plant uptake. So to set up the design a little bit further, a reminder, we have 6 populations, 3 are from loam origins and 3 are from sand origins. And so from each one of these populations, we harvested 10 maternal families. And then of each maternal family, we germinated 8 individual seeds, and we did that twice because we have 2 treatments, which I'll mention in a minute. But for now, it's just to say that we have a total of 960 plants that are growing. And so all 960 seeds were grown in a randomized block design, and we built special growth boxes for our seeds that were separated by these grids. And then we placed our growth boxes into a growth chamber, and we controlled for how much light they got, the temperature, and the humidity. And then for the treatments, we manipulated the amount of water seedlings received, where one treatment is well watered and the other is water limited. And I want to point out that we chose water limitation to mimic what the plants may experience with climate change in the Great Plains region. And so as a reminder, precipitation patterns are expected to have large extremes, including flooding and drought. However, because temperatures and the evaporation demand are expected to steadily increase, we know that there is going to be less water in general across the landscape. So for roughly the first 6 weeks of growth, all seedlings grew under the same controls and were maintained under well-watered conditions. And then to maintain the amount of water between each treatment, we calculated the pot capacity using the gravimetric method. And so the gravimetric method is simply where you take the soil medium the plants are growing in, and you completely saturate it, and then you completely dry it, and then you find the difference, and then you divide it by the difference, and you multiply it by 100. And so with that method, the well-watered conditions were maintained at 90 to 100% pot capacity. And then after 6 weeks of growth, the seedlings were large enough to initiate the treatments. So half of the seedlings stayed under those well-watered conditions, and the other half were subjected to about 50% pot capacity or the water limitation treatment. And so for this experiment, we measured a lot more traits, and all of them are either directly involved with the balance of carbon uptake to water loss, or they are the end result of that balance. And so for this talk, I am specifically going to focus on water use efficiency, like we saw in the last chapter, as well as the root to shoot ratio of our plants. And so specifically, we're asking, is there an effect of soil texture origin on trait response to changes in water? And then we also want to know if traits are under genetic control, environmental control, or if they're under both genetic and environmental control. So for our expectations of soil texture, under the well-watered conditions, we expect that loam populations are going to have a greater water use efficiency and a greater root to shoot ratio relative to sand. And for the water limitation treatment, we expect the opposite, right? Because populations sourced from sand soil textures have potentially adapted to compensate for a limited water supply. So we expect populations that originated from sand will have a greater water use efficiency and a greater root to shoot ratio relative to our loam populations. All right, so in this figure, you're looking at the mean soil texture response under both treatments, and it's split by soil texture origin. And so on our x-axis, we have well-watered conditions and water-limited conditions. And then our y-axis, we have our carbon isotope composition, where again, up here is high values of water use efficiency, and then the more negative the carbon isotope composition is, the lower, indicates a lower water use efficiency. And so I'm giving it away at the top, but the first thing to note is that water use efficiency between or across treatments was different, okay, where water use efficiency was higher in the well-watered treatment. However, when looking at the influence of soil texture origin on these trait differences, within treatment, we did not see any effects. So our expectation that loam would do better than sand under well water was not met, and our expectation that sand would do better than loam under water limitation was not met. If we look at the root to shoot ratio of plants, we again, we're seeing differences between treatment or across treatments, excuse me. But this time we see that soil texture origin does impact the root to shoot ratio, but only under well-watered conditions. And we don't see soil texture origin influencing trait response while under water limitation. So under water stress, soil texture was not impacting trait responses. So we have investigated briefly the influence of soil texture on our traits. And so now we're going to look at the plasticity of these traits, specifically whether they're genetically controlled, environmentally controlled, or a combination of both. And so if they're under genetic control, we expect that within one environment, we're going to see population level differences, but their response across treatments is going to be the same. And so this slope of reaction is going to be 0. If these traits are environmentally controlled, we expect the opposite. So we expect that there's not going to be population level differences, but they have a slope of reaction where this trait is changing across water environments. And then finally, there could be a gene by environmental control where there's clearly genetic differences between our populations, and then the slopes of reactions are different for each population. So we're going to start with water use efficiency once again. So shown on our x-axis, again, we have the well-watered treatment and the water-limited treatment, and then we have the carbon isotope composition on the y-axis. And so looking at this graph, it looks like some populations have a steeper slope than others, which could potentially indicate a G by E situation. However, due to that variance surrounding our population means, we did not find genetic differences between our populations for water use efficiency, and we also didn't see it change across water environments at the population level. Unlike water use efficiency, we could pick apart whether traits are genetic, environmental, or genetic and environmental control. And so here we can see that populations have differences in their root to shoot ratio within each treatment. So traits are genetically controlled, and we can see that each population's root to shoot ratio changes across treatment. And so populations exhibit a plastic response as well. However, because the slope of reaction is pretty much the same for all these populations, we're not seeing a G by E interaction across populations. All right, so to wrap up that brief chapter, we investigated the effect of soil texture origin and how that influences trait response to water limitation. And we found that soil texture does not matter for water use efficiency. We did find an influence of soil texture origin on root to shoot length. However, it was only under well-watered conditions, and we didn't observe that effect under water stress. And so understanding how selection acts under fine scale differences is important when we consider if we want to move seed across soil textures. And so what we found is that if you want to move seed to drier conditions, soil texture doesn't matter for root to shoot ratio. However, it may be important if seed is moved into the locations without water stress, and it may be different for other traits. We also observed that whether a trait is genetically controlled, environmentally controlled, or if it's a combination of both, is trait specific. And so for water use efficiency, we didn't find differences in our population water use efficiency regardless of treatment. But for root to shoot ratio, we found both genetic differences and plasticity in the trait. And so because we saw genetic differences in source populations, we can say that where we source our seed will impact the root to shoot ratio, and that plasticity in root to shoot ratio is important for restoration because that potentially, again, can potentially buffer populations against changes in climate. All right, so summing it all up, for restoration, where you source your seed matters. And it matters because selection associated with your source environment can influence the distribution of genetic variation in traits important for adaptation. So for prairie smoke, the amount of genetic variation matters because it tells us the risk of transfer. So when the alvar populations moved into the prairie environment, we saw that they managed their water use better than the local prairie populations. But we need to also think beyond just what the transfer risk is and think about how plants will adapt to climate change, and the distribution of variation can give us insight into that. And then lastly, we saw that selection influences traits differently. So teasing apart trait correlations or determining which trait is more important for persistence on the landscape is going to be essential for population persistence in the future. So I want to thank funding, and I want to also thank the people behind that funding. So my advisors, Jill and Marissa, and then my committee members, Dr. Osorno and Dr. Laura Aldrich-Wolf, everybody in the Hamilton lab, Wendy and Jeff, Nina and Paula, and then everybody else who helped me collect data, clean seed, I should say. It'd be nice if they came and helped me clean my house, but nope, I had to do that myself. But then pretty much everybody, it's just been a wild year for people. So I think a lot of support has been happening, and it's been greatly appreciated. So if you see your picture up there, extra kudos to you. And with that, I'll take any questions.
Thanks, Kate. Very nice job. Very nice job. I guess I'll start off with one question. From your first chapter, I noticed that there's really high variation in your heritability estimates, but your evolvability estimates were virtually no variation at all. Mm-hmm. And so I was curious about that. And also that there's actually, I thought about, there's this idea that if a trait or that in cases where you have high plasticity, that it actually may facilitate evolution because you have phenotypes that are in the new extreme, at the extreme end of the tail. And so even if there's low genetic variation in that trait, that heritability might facilitate evolution. Now this is different though, and so maybe you could comment on that if you understand my question. I'm sorry.
Yeah, well, so for the first part, you don't see variance around evolvability because this isn't necessarily meant to be shown as a figure. And so the evolvability and the heritability are both taking the additive genetic variation, but it's doing different things with them. And so by using evolvability, we're also saving the variance for the heritability component. It's a little confusing, sorry. But basically there isn't a variance component surrounding evolvability because it's standardizing it in a different way. And yeah, and then the second part of your question, you're saying plasticity could potentially facilitate adaptation or evolution. And I don't think that's wrong. I would have to like think about that a little bit more for this study. Yeah, can you repeat it a little bit so I can think about it a little bit more?
That's okay. That's okay. I think I'm down in the weeds. I can talk with you about it later. I see there's a question from Lydia.
Hi, great job on your presentation.
Thanks. Oh, you muted yourself. I'm not sure if you meant to.
Can you hear me?
Yes, I can hear you. Yep.
Okay. So I'm in the geology department. Whenever I hear someone give a presentation that includes a stomatal index, you know, this is one of the most widely used methods for understanding carbon dioxide atmospheric concentrations in Earth's past. But there's much less control on like different species, on source materials. Do you think the stomatal index, when you have less control on species information, is a useful proxy in fossils when it's basically you can't improve on it that much? Like, do you think that it's useful in that way?
Oh, kind of outside of my realm, but I think if maybe it could be useful if like the fossil is still alive. Like if prairie smoke was a leaf of prairie smoke was fossilized however long ago, and that plant is still alive today, and you could make that's hard. I'm not sure about the fossil, but for my project, I'm interested in how big the stomata are. And so I think like if you saw evidence of different stomatal sizes in the same species, it would probably still mean the same thing I'm seeing, where that smaller stomata can respond faster to the environment, but I have no idea how the different, the historical amount of carbon dioxide would influence that. Sorry, I'm out of my ballpark here answering this question.
No, that's okay.
I'm not sure. That's really
you bring
cool,
up
though.
stomata. I'm interested in this proxy, and I know that there's some problems with it. So I definitely I appreciate that perspective. Thanks.
I want to see some fossils of stomata now.
I'll send you stuff.
Cool.
Other questions? You can put them in the chat or just start speaking.
What's the pandemic Lovato? That's a pandemic.
Hey, I have a question.
Hi, Laura.
Hi there. So maybe I missed it. Maybe you mentioned at the beginning, but you're using seeds from different sources and then either in the common garden or the greenhouse. So what did you do to control for maternal effects? Or are those just folded in there? Can you talk a little bit about that?
So for like on the screen right here, the estimates of heritability and evolvability, we multiplied our ratio of VA over VP by a coefficient of relationship to account for both our half sibling design, which I didn't talk about in the common garden, and then also to account for the selfing that prairie smoke can undergo. So to get these estimates, that's how I controlled for the maternal effects. And then in the other chapter, I haven't yet. There's some other analyses I want to do. As you can see, that one was a little bit shorter. I'm not quite done. So maybe I'll send you an email and we can talk about how I can control for the a little bit better in the second chapter.
Yeah, so then along those lines, did you see, since it sounds like you did take that into account in your experimental design, did you see any evidence for maternal effects?
So at one point, I've looked at this a lot of different ways already, but in that second chapter, at one point, I did split my reaction norms by population and just looked at the maternal response. And there wasn't a whole lot of difference. So like within a population, each maternal family basically did the same thing. Is that what you're asking?
Yeah, I mean, I was just kind of curious just to try to unpack a little bit the ways in which you were measuring and thinking about heritability. So that was helpful.
Yeah, I haven't like really visualized the family structure of heritability here. I know that for some of my heritability estimates for Manitoba, they're like nothing because I only have 2 populations. And so my family structure is a lot less compared to the other 2 regions. And that's probably why I'm seeing a lot more variance around the Manitoba estimate in this specific graph. But yeah, definitely something to think about, especially if I want to start doing these kind of analysis for my second chapter too.
Yeah, and it would be interesting to think a little bit about, right? Like, I mean, you're thinking about stomates in the context of water use efficiency, but another component of that, right, is thinking about carbon dioxide concentration, right? And what that means in terms of stomatal conductance, right? So basically, plants can get away with, right? Actually, when there's a higher concentration of carbon dioxide, they can get away with less aperture on their leaves, basically, in terms of stomates.
Because they don't have to be open as long to collect the same amount. Yeah.
Right?
I think, yeah, that makes sense. I think like, oh, go ahead.
No, I was just going to say, I mean, getting back to Lydia's question, right? That is one issue is you need to know what the carbon dioxide concentration is in the atmosphere. When you're right, you're looking at the patterns.
I definitely think that's important. And I think, but like in this case, they're all experiencing the same amount of CO2 in the atmosphere because it's in the common garden. But that's something definitely to think about for like future projects if I were to like do a reciprocal transplant with this. And then if I wanted to compare it like that, for sure.
Well, right. And what I meant to get at a little bit there was that I think it's important to think about the fitness consequences, because having higher water use efficiency when carbon dioxide concentrations are relatively low in the atmosphere, right, potentially requires a trade-off so that you're a smaller, slower growing plant. That should be less true as the carbon dioxide concentration increases.
Absolutely. Yeah.
So I think it's interesting to just think about your data a little bit in that context in terms of like thinking about the future, and right.
Yeah. Absolutely. Yeah, absolutely.
Nice talk.
Thanks.
Excuse me, Kate. Hi, nice job. David Hopkins here. Kate, in this picture that you're showing right now, that shows the heritability and evolvability, the camera angle is quite interesting. From my experience, generally, this sort of seems like a bit of a prostrate little form. I've seen a lot of old man's beard or geom on the prairie, and in the sandhills, of course, there's a lot of competition. Did you see much in your reading about different sort of, as it were, ecotypes in terms of habit of the geom from New York in through the prairie, either in terms of height or leaf thickness, or I would think our prairies, the alvar was interesting. Thanks for a new word. I appreciate it. The prairie is pretty rough. You were talking about the reservoir, the soil, which makes a lot of sense, but it still, as you well know, can be pretty hard to cope with. Any differences in leaf thickness or degree of leaf thickness, anything along those lines that would help explain how the leaves shape or thickness might vary besides stomates from New York, say, to
Mhm.
Idaho or Western North Dakota.
So yeah, so this part of my project is a continuation of this long-term common garden study. And so Zeb Yoko, who I'm not sure if is still here, actually did a whole bunch of other data collection, and he did look at leaf morphology. I could not tell you off the top of my head how they were different, but I think he did find differences in leaf morphology. I'm not sure if he didn't look at, I don't think he looked at thickness. I think he just looked at the shape and how loby leaves were. So I can't tell you if Alvar was lobier versus prairie, but I can tell you that they did look different.
Interesting. Yeah.
Yeah.
Thanks very much.
Thanks for coming, Dr. Hopkins.
I love plants. You know that.
Yeah. A little bit of soils in here. Not a lot, but a little bit.
Is there any more questions?